Hello and welcome to Taconix's webinar titled Discover huNOG-EXL, Extending the Limits of Engrafted Human Immune System Models, presented by Dr. Michael Seiler. Please allow me to introduce myself. My name is Megan McBride and I will be your moderator today. I work in Taconix's Product Management Group where I am responsible for commercialization of animal models. Before we get started, I'd like to address a few practical matters. This webinar will be made available to all attendees. We will email a link to the presentation to you by the end of this week. Directly after the presentation, Dr. Seiler will answer questions from the audience. So if you have a question, please type it in the question box in the control panel. The other attendees will not be able to see your question as it is sent directly to Dr. Seiler and to me. If you cannot see the control panel, just single-click on the single arrow at the top of your screen. In case your question is not answered at the end of today's session, we will respond to you within the next 24 hours via email. If you are experiencing technical difficulties, please send the host a chat message or raise your hand and our staff will be able to assist you. So, let's get started. We hope you will enjoy today's webinar about the huNOG-EXL model. Our speaker, Michael Seiler, received his PhD from Baylor College of Medicine. He did his postdoctoral training at the University of Chicago with research into natural killer T-cell lineage commitment. He is currently Associate Director of Product Management at Taconic. Dr. Seiler.
Thank you, Megan, and thank you, everyone, for joining this webinar today. I hope to spend the next few minutes taking you through a narrative of the history of the immune-deficient mouse model as a host organism for immune system engraftment, with specific respect to the NOG or the NOG mouse, developed by our colleagues in close partnership and close partners at the Central Institute for Experimental Animals in Japan. I will talk also about the overviews, strengths and limitations of current immune system engrafted animal models, methods that are being used to exploit the human immune responses represented in engrafted mice for immuno-oncology research, and we'll spend a few minutes talking about the huNOG-EXL, the new mouse model recently developed by CIEA, as it imparts some significant improvements in human immune cell lineage commitment that we will focus on for the duration of the talk. The huNOG-EXL has very interesting and cutting-edge applications in allergy, inflammation, and cancer biology. First, a word about the Central Institute for Experimental Animals. Taconic and CIEA have an ongoing partnership to develop and produce some of the world's leading animal models for immune cell research. The Central Institute for Experimental Animals was founded as a nonprofit organization in 1952. It is located in Kawasaki, Japan. It employs over 100 researchers and technicians, including visiting scientists. Their mission statement is to develop advanced animal models to further the development of medicines for human health. The origin of the CIEA NOG mouse began in 1985 with the first development occurring between the cross of the skid mouse and the NOG mouse. The combination of these two mice imparted properties of immune deficiency that were exploited for many years. Later, the independent identification of the common gamma chain of the IL-2 receptor and its implications in immune biology were combined to develop the NOG or the NOG mouse. This decades-long process has yielded an animal model that is quite suitable for immune cell engraftment. The NOG mouse, the nomenclature shown here on the slide, is running up the slide in a diagonal pattern. It highlights some of the important intrinsic biological qualities of the NOG mouse strain. The NOG background itself imparts a polymorphic signal regulatory protein alpha, which I'll spend a few minutes talking about in the next slide, that is quite important for the ability of this mouse to host immune cell engraftment. The NOD background is also complement C5 deficient and has demonstrated impaired dendritic cell and macrophage function in phagocytosis and antigen presentation. The SCID mutation, as a consequence of the combination of the NOD-SCID background, is a disruption in a critical enzyme for DNA recombination. Recombination is a critical step in T and B cell VDJ recombination, and therefore the disruption of this gene results in poor lineage commitment in B and T cell sublineages from the common lymphoid progenitor. Further impairment of the immune system is communicated by the targeted disruption of the IL-2 receptor gamma, or common gamma chain of the IL-2 receptor as it's known. As the gamma chain is critically important for IL-15 mediated signaling, and in NK cell developmental biology, the NK cell portion of the immune system is completely devoid in a gamma chain disrupted animal. This is well-established biology in many iterations of gamma chain disruptions, and is one of the critical factors associated with the immune deficiency evident in the CIEA NOG mouse. I mentioned that signal regulatory protein alpha polymorphism evident in the NOD background, and this has been linked strongly to the differences in hematopoietic stem cell engraftment associated with the NOG mouse. On the bottom of the slide is a series of flow cytometry plots that show the opsonization of mouse SERP alpha on immune cells with a soluble version of human CD47, a cognate receptor that communicates an anti-phagocytic signal to the host. The increased binding affinity of the SERP alpha evidence in the NOD background is compared to BALP-C and C57B6 mice. As you can see, the high affinity shown in the binding kinetics on the top right-hand part of the slide is clearly in favor of the NOD strain. This translates to a significant increase in the ability of a similarly attenuated immune system mouse on a NOD background, BALB, or B6 strain animals to accept human hematopoietic stem cells. Shown in the top left are relative rates of engraftment measured by the percent chimerism of human cells in peripheral blood of mice engrafted with hematopoietic stem cells in NOG, BALB, or B6 mice of the aforementioned similar disruptions in DNA repair and gamma chain. Clearly, the NOG background is superior to the other strains in its ability to accept human hematopoietic stem cells, and this has been further characterized and related to the NOD SERP alpha polymorphism. Before we dive into the differences in sublineage commitment, I'd like to spend a moment setting the background for just how an immune system model is developed in vivo. This is accomplished by the engraftment of various cells and tissues that are implanted or injected into an immune-deficient mouse and allowed to establish some functional activity. The measures of success for this that are commonly represented and that will be presented throughout the presentation today are the presence and quantity of human leukocytes evident in blood, typically measured by flow cytometry. Additional aspects of functional activity of the immune cells in the blood can also be performed, and in some cases will be displayed in the data of the presentation. There are different models for different uses. This is a very important point to consider as you select different models for the preferred outcomes. We understand that there is no pure recapitulation of human immunity in a rodent system. And in that sense, these models can be purpose-built for specific applications related to the presence of human immune cells in a living system and the ability to exploit their mechanistic functions. To be sure, there are different models for different mechanisms of action, and today we will cover both the NOG and the huNOG-EXL as different models associated with different properties. The ability to engraft immune system into immune-deficient mice can take multiple routes and strategies. Shown here are the three most common methodologies for engraftment procedures in use today. The most common, shown on the top of the slide, is essentially independent of strain, but does include the intravenous injection of purified CD34 positive hematopoietic stem cells. The immune cell types that are evident in the blood of these mice include T and B cells, NK cells, macrophages, granulocytes, and have applications across immuno-oncology, the basic discovery in hematopoiesis, T-cell responses, and tumor infiltration by lymphocytes. Shorter-term experimental models include the engraftment of peripheral blood mononuclear cells, typically engrafted in older animals with a relative lifespan of about 30 days. These mice succumb to graft versus host disease, but can be exploited for specific immune modulatory effects of small molecule compounds and monoclonal antibody therapies, as well as vaccine and infectious disease responses. The BLT procedure for engraftment of immune system upon immune deficient animals includes co-implantation of liver and thymus tissue fragments under the renal capsule, combined with the intravenous injection of purified enriched autologous CD34 positive hematopoietic stem cells. There are nuances to the biology associated with this combined liver, thymus, and stem cell engraftment strategy that include the T-cell selection on the thymic organoid, yielding T-cells specifically reactive to human HLA program. Over the last several years, improvements in immune-deficient host mice have paved the way for unique human hematopoietic stem cell xenograft mouse models that support the development of human hematolymphoid systems. The first iterations of this work were done in NOG-SCID mice and have progressed over time in their complexity. Over time, the utility of the host system has increased, and the knowledge gained by examining human immune cell lineage commitment has helped to define the hematopoietic stem cell hierarchy, differentiating in these living systems. Important for the successful engraftment and establishment of human immune cells are three major factors. These include the tolerance by the host mouse, the appropriate residence in immune cell niche microenvironments, and identifying limitations in cross-species interaction with critical lineage commitment factors such as cytokines, chemokines, and chemotactic signals. The cartoon on the slide displays some of those interactions that take place and the lineages and the subsequent sublineages as effector and innate immune cells develop and differentiate. Hematopoiesis, to be sure, proceeds through an organized developmental hierarchy, initiating with a hematopoietic stem cell that can give rise progressively to more committed progenitors and eventually terminally differentiated blood cells. When transplanted into the mouse, the HSC develop and differentiate through the complex communication of both membrane-bound and soluble molecular signals that are largely interdependent on the host to drive lineage commitment. While this slide shows some of those interactions, the interdependence of host-derived factors on human HSC differentiation is complex and a significant area of active study. The next few slides will outline some of the advances in understanding the processes and outcomes of HSC engraftment in the NOG mouse and the additional advances to overcome cross-species limitations. The most common method, as I mentioned, of reconstituting the human immune system in mice is by administering human hematopoietic stem cells. These models can generally be described as excellent models of T-cell function, exploited in research programs investigating immune checkpoint inhibitors and other T-cell mediated mechanisms of drug activity. The process to engraft a mouse with a NOG mouse with human hematopoietic stem cells is shown on the slide here. After a preconditioning irradiation step and a dwell time of approximately 1 day, 3-week-old NOG mice are engrafted with 100,000 CD34 positive HSCs via tail vein injection. The animals are left to develop and differentiate the immune system for about 12 weeks before the first typical quality control assessment is made. It should be noted that this 12-week timeline is arbitrarily chosen to identify at the soonest time point the majority of animals that have successfully developed and differentiated into chimeric animals. We measure this by assaying the peripheral blood and preparing it for flow cytometry, staining for leukocytes of mouse and human origin as shown in the middle of the slide. Our metric of 25% or greater is also chosen as a suitable outcome that defines an animal that has been successfully chimerized in this process. Once established, these chimeras are quite robust and will live for greater than 1 year. At Taconic, we typically ship these animals between 12 and 24 weeks post-engraftment once they have reached their QC positive level. If we dig deeper into the biology established in the NOG mice upon hematopoietic stem cell engraftment, we see that the ratio of B and T cells is inverted compared to what is normally identified in peripheral blood of humans. This is a common hallmark of immune system engrafted animals and is also evident on the slide in the pink box in the middle. Nevertheless, the successful engraftment of greater than 80% of the animals attempted is shown here on a sample set of 91 huNOGs, all prepared from the same donor lot of hematopoietic stem cells. One question we often receive is how consistent is the donor-to-donor variability associated with immune system engraftment. Shown on this slide is about 1,000 huNOGs generated from 12 different lots of immune cell donors. What you can see is that the majority of animals engrafted will reach the 25% or better engraftment level by 12 weeks. Again, I stress that this is a temporal kinetic process for evaluating immune system engraftment, as the immune system will continue to grow and differentiate in these animals, reaching a plateau of around 35 to 50% in animals that have been successfully chimerized. We again choose 12 weeks as an important metric to determine whether the chimera has been established and the animals are suitable for study. There is one donor lot evident identified by the red arrow in the middle of the slide that completely failed to engraft, and this happens with some periodicity. Also notable is the group of animals closest to the y-axis, where about half of the animals have reached the 25% or greater metric, and half lie just below it. Those animals measured at a time point subsequent to 12 weeks will invariably reach that greater than 25% mark, and those animals will be remeasured within a few weeks. Given the propensity of these mice to develop and differentiate into lymphoid lineages, these animals have been exploited heavily in immuno-oncology research. As the immunologic synapse contains a number of immune checkpoint inhibitor targets of active interest in drug discovery, T-cell an increasing role in evaluating these models for their suitable efficacy in combined immune system and tumor engraftment strategies. An important question to answer with respect to whether an immune system engrafted animal is suitable for combined immuno-oncology research is whether a tumor can grow in the presence of the tumor similarly to how it does, presence of an immune system similar to how it grows in the immune deficient mice. To ask these questions, we engaged a company very active in the patient-derived xenograft commercial space called Champions Oncology. Champions Oncology is very interested in personalized medicine and the application of patient-derived xenograft work to identify therapeutics specifically targeted to tumors of specific subtypes. In this experiment, Champions Oncology engrafted non-small cell lung carcinoma from their patient-derived xenograft tumor bank into both humanized and immune-deficient animals. And what was clear is that whether the immune system was present or not, the tumors grew with similar efficiency. In further work, they showed that the immunotherapy ipilimumab, a target of the T-cell molecule CTLA-4, an important communicator of a suppressive signal to T-cell populations, could be targeted and successfully activated in both an efficacy outcome in tumor growth as well as specific mechanism of action on T-cell activation when combined in an immune system engrafted NOG mouse and tumor graft strategy. What they found is that human hematopoietic stem cell engraftment resulted in long-term survival of functionally competent immune cells in vivo, and that patient-derived xenograft tumors grow similarly in immune system engrafted mice compared to unengrafted immune-deficient animals. There was measurable T-cell activation and anti-tumor immune responses after treatment with ipilimumab. However, we know that this immunologic synapse has a number of cellular partners that participate in the communication of immunity against tumor antigens. These include antigen-presenting cells of dendritic cell and macrophage origin, in addition to the T-cell component. Therefore, there is a need for animal models that support sublineage differentiation into the myeloid family. There has been a significant advances in myeloid lineage differentiation predicated on the biology of these immune-deficient mice, in that one major consequence of the increased adoption and understanding of immune-engrafted mice has been that key limitations in myeloid lineage commitment are associated with poor cross-reactivity of human cytokine receptors with host mouse cytokines. Prior work in this field has shown that cytokines GM-CSF and IL-3 in specific, delivered as recombinant proteins, gene therapies, or via genetically engineered into the mouse, into the host system, are sufficient to rescue myeloid lineage differentiation. Consistent with this, our colleagues at CIEA have developed NOG mice transgenically expressing those two key cytokines required for myeloid lineage commitment. The resultant mouse is a suitable host to support both lymphoid and lymphoid B and T cell lineage commitment, as well as innate immune cells of myeloid lineage family. These include macrophages, basophils, neutrophils, mast cells, eosinophils, and monocyte lineages. In late 2013, researchers at the Central Institute for Experimental Animals published the first manuscript describing the immune system engraftment of the GM-CSF IL-3 double transgenic NOG mouse, heretofore referred to as the huNOG-EXL. What they showed is that there was an improvement in overall cellularity, both evident in the gross anatomy of the spleen, shown on the top right of the slide, the enumeration of human cells in comparison to non-transgenic animals, and the temporal increase in relative chimerism, shown in the middle of the slide over time. Our experience at Taconic has been similar to the data shown by CIEA, whereby the chimeric ratios evident in immune system engrafted lots of huNOG-EXL mice showed significant levels of chimerism that remained stable and suitable for experimental use well beyond the 24-week time points. We've also done direct head-to-head comparisons at early time points post-engraftment, comparing donor cells from two different donors engrafted into either NOG or NOG-EXL mice. What was clear is that in both iterations, the NOG-EXL mice differentiated to chimeric ratios to a higher degree than their NOG counterparts. So the same cells into two different strains of mice yielded different chimeric ratios, exemplifying the ability of these mice to support greater cellularity and lineage differentiation. The immune system engraftment with different sources of CD34 positive hematopoietic stem cells is an additional common question that we receive. To address this, we engrafted the huNOG-EXL mice with either umbilical cord blood derived CD34 positive hematopoietic stem cells or liver derived CD34 positive hematopoietic stem cells. In both cases, sufficient chimerism was established in the majority of animals, with most of them reaching a chimeric ratio at 12 weeks of between 40 and 60%. The sublineage differentiation included the improved characteristics of myeloid and monocyte lineage differentiation, shown on the slide in the top of the slide of the huNOG-EXL compared to the non-transgenic. The conclusion here is that the transgenic support of GM-CSF and IL-3 can induce myeloid lineage commitment and increase the frequency of both myeloid and monocytes in the blood of huNOG-EXL mice. Further characterization of granulocyte differentiation is shown by purification of large granulocytes by fluorescence activated cell sorting and magenta staining. Eosinophils, basophils, and immature neutrophils were evident both by their shape and granularity and their staining profiles, as shown here. Moreover, macrophages differentiate and develop in greater frequency in the huNOG-EXL compared to the non-transgenic NOG mice. Those cells, once differentiated, home to tissues where we would expect to find them, including lung, liver, and spleen, and take up residence within those tissues. So the frequency and presence in peripheral tissues of immune cells has been shown in the huNOG-EXL mice. To further ask the question whether those cells are functional, the researchers at CIA performed the passive cutaneous anaphylaxis reaction, where animals were preconditioned with soluble IgE and challenged with NT-based, BSA-based protein challenge. This reaction elicits the extravasation of Evans blue staining and can be monitored in a macroscopic way by evaluating the skin for the extravasation of the dye. What is shown here is the difference in the PCA reaction in the huNOG-EXL compared to the non-transgenic animals, as well as mice that were non-humanized, showing that it was not the extravasation of the dye was not related to mouse mast cells. Additional interest in the development and differentiation of myeloid lineages is related to the recapitulation of tumor microenvironment. If the host system can support the development and differentiation of these sublineages, one hypothesis is that these cells can subsequently home to tumor xenografts, take up residence, and impart some of the suppressive characteristics evident in clinical specimens. In preliminary work performed by the Central Institute for Experimental Animals, they have observed the induction of tumor-infiltrating suppressive myeloid cells in the NOG GM-CSF IL-3 transgenic, also known as the NOG-EXL. To summarize the data shown at this point, the huNOG-EXL mouse shows significant improvement in myeloid lineage reconstitution and overall cellularity. It has, by supplying the critical cytokines GM-CSF and IL-3 off of transgenic expression, there is rescued granulocyte and macrophage differentiation and function. There has also been observed differentiation of myeloid-derived suppressor cells, which has the potential uses in cancer immunotherapy studies. Active investigation into these key areas of both drug discovery and basic science include allergy, inflammation, innate immunity, and cancer. I will take this last moment to mention the upcoming international workshop on humanized mice, where advances in model development and sublineage support by cytokine administration and expression will be detailed, and further applications will be disclosed. And finally, if you would like to learn more about the immune system engrafted models currently offered by Taconic Biosciences, or to read related publications on the NOG mouse, please navigate to our websites for either the NOG or the huNOG-EXL. If you would like to schedule a consultation or request pilot pricing information, please reach out to us through the info@taconic.com website. Live inventory are now available for engrafted mice, and so your piloting and discovery sciences to support your research program can be accommodated, so please contact us.
Thank you, Mike, for that excellent seminar. We are now accepting questions from the audience. Please go ahead and type those into the question box. Again, only the presenter and I will see them. The other attendees will not. We have already received several questions, so I'll go ahead and start. The first question we received, in the slide where you show percent human CD45 cells over time, the huNOG-EXL might show chimerism at 4 weeks post-engraftment. What advantage, if any, does that confer to studies in immunology or oncology?
So that's a very good question. The one key observation that was noted by the researchers at CIA and has been confirmed at Taconic is that the advantage of cytokine expression does result in identifying successfully chimerized mice much sooner in the process. The typical strategy of determining chimerism is based on when human cells are most evident within the blood to a sufficient degree that we can score them by flow cytometry. This has typically been done at a 12-week post-engraftment timeline. But what is clear from CIEA's data and our data as well is that chimeric establishment can be detected as early as 4 to 6 weeks post-engraftment. That timeline is advantageous to the researcher in immuno-oncology if they wish to combine this with a slow-growing patient-derived xenograft tumor where they can begin the engraftment process much sooner post-engraftment.
Thank you. Our next question is, let's see here. It's about how the huNOG-EXL was made. So the question is, how are those transgenes expressed? Were they made transgenically through pronuclear injection, or was it a knock-in? And if it's the former, what are the promoters that were used?
Yes, these transgenes are expressed off of an SV40 promoter, viral promoter. There is a single insertion event of 2 independent transgenic cassettes. The levels of transgenic cytokine expression are detailed in the manuscript published in the Journal of Immunology by Dr. Takeshi Takahashi I think a year or so ago. The levels are in the double-digit picogram range, if my memory serves, and that's an important point to note because the expression levels of cytokines can result in chronic supraphysiologic levels that may have influence on downstream immunologic function. What we do know is that whether the cytokines are administered either as recombinant proteins expressed off a transgene in sufficient quantities, they can push myeloid lineage reconstitution.
Okay. Our next question is about comparing some of the other models that are out there. How does the GM-CSF IL-3 NOG compare to some of the other cytokine transgenics? So the NSGS or the Mr. G are common models?
Yeah, this question is related to the last question, and I think there are a couple key points to note. The similar biology reflected in either the systematic knock-in of human sequence, as was done in the Mr. G mouse, or the transgenic expression associated with other transgenes under different promoters, or recombinant protein administration. In each case, 2 cytokines have been consistent. That's GM-CSF and IL-3, and it's related to the the close proximity to which those coding sequences reside in the genome. In both cases, the expression of those cytokines has been sufficient to push myeloid lineage differentiation in all 3 mice. However, the downstream consequences of cytokine concentration in blood may have impact on the overall survivability of hematopoietic stem cell engraftments, and as well as downstream immunologic function, whether it be in the context of tumor immunology or in basic science of immune function.
Thank you. Our next question is about the PDX experiments that you discussed. When doing testing in the PDX models, are the hematopoietic stem cells obtained from the same patient from which the tumor tissue was derived?
So in this instance, no, they were not. That is a question that bears some deeper explanation into the source material associated with hematopoietic stem cells. In order to accomplish the system where a truly autologous tumor and immune system combination is reestablished in the mouse is exquisitely difficult, because it would require hematopoietic stem cells be obtained from the specific patient. So in these instances, what we're evaluating are immune system donor hematopoietic stem cells that were originally HLA type to at least the HLA A2 group level, and matched to patient-derived xenografts of HLA A2 group level also. So the immune system shared at least one allele of HLA A2.
Very good, thank you. It looks like that is all the questions that we have received, so we will close the webinar. Thank you again for attending, and thank you, Dr. Seiler, for the excellent talk.